which is very nice. Before starting, I want to point out that some of the data is also acquired by Michelle Digman. Okay, so the number and brightness is about, well, you can measure the numbers, so the concentration, which you can also get from the RIGs or from the FCS. But yeah, as the second word in this method, brightness, describes, we can look at brightness and thereby get the aggregation state. If two fluorophores are moving together, they should on average have to double the brightness. So from the brightness, we can tell if they are moving together or not. And you can do this, of course, for proteins in cells or other molecules in cells, lipids in cells, which can be very powerful. Okay, so one thing you can do if asked, well, what is the concentration in a particular region in the cell? Of course, you can look at the intensity. Let me change this pointer here. So, for example, we are here in spot A and we get some kind of average counts per second, let's say 30,000. And then we go to another spot where we have a much higher intensity, whatever, 90,000. And then if we do a reference measurement with a solution, in this case of the green fluorescent protein, where we know the concentration and the count rate it gives, we can infer the concentrations in those regions of the cell. So in this case, region A, for example, would have 10 nanomolar and region B would have 30 nanomolar. And this accurately describes the number of molecules that you have in those regions. However, what this doesn't tell you is if those molecules, are they bound together or are they just accumulate in this region because they are contained in some larger structure or something. So we cannot really measure the aggregation state just by looking at the intensity this way. There are other methods you can do, which is, for example, first resonance energy transfer or FRET, where you have two fluorophores, and they can, if they're close together, there can be energy transfer between the two, either a homofret if it's the same molecule, or if you have a donor and an acceptor molecule, you have transfer from the donor to the acceptor. That only happens if those two molecules are very close, within a nanometer or so. And then you can conclude that the two proteins that those fluorophores are attached to are bound together. So this is a very sensitive method and very nice method, but I'm not going to talk about it today. It's very sensitive to the formation of pairs. But what about other, for example, higher states of aggregation? Another method you would be able to look at that is image correlation spectroscopy. Of course, if you have very large aggregates, this will give you an increase in the size of your spatial correlation, and you can get the aggregation state from that. But that typically only works if molecules are moving slow and if they're relatively large. Okay, so what can the number and brightness method do for you? So it can provide you with a pixel resolution map of molecular, the number, so concentration, and the aggregation state of the molecule of interest in cells. How does it do this? Well, we look at the first and second moments of the fluorescence intensity distribution at each pixel. So this means we need to have more than one image. Another reason, when you acquire the laser scanning image, yeah, we have to accumulate 100 or even more images than that. What are the typical source images, we can have raster scanned images like from the RIX experiment. But for the number and brightness, we can also have camera images from like a total internal reflection microscope or a light sheet microscope that were acquired with fast cameras. Because here we don't need a time structure within the same image as for the RIGs. We just look at the statistics in one pixel from one frame to the next. So here we can also use camera images. What do we get? We get the number and brightness maps. So pixel resolution map, and you get the 2D histogram of the brightness versus intensity, which can show you if you have these aggregations, you can select, for example, only dimers from that and then map it back to your image to show you where they are in the cell. You'll see an example later. So you have this kind of cursor selection method, which is very convenient, used in a lot of things. Also, I think during this week you will hear about the lifetime and the phases. So that's very nice where you have those two plots where you can select a certain property and then show you where it is on the image. If you do the measurement, you need to take into account what kind of detectors you are using, and I'll explain that to you in the following. And again, on Friday in the practical session, we're also going to look at NMB data and how you have to correct for your detectors. Yeah, and we'll show you how to get the data in and analyze some example data. Okay, so the question is, how do we distinguish pixels that have many dim molecules from pixels that have fewer but very bright molecules? Because this can potentially give you the same intensity. Many dim molecules can give you the same intensity as few bright molecules. So if we look at the average, and we have few bright molecules, in this case, for example, we have an average count rate of 19,000 counts per second. Here we have on the right-hand side, we have many but dim molecules, and we can have exactly the same intensity. So the average in this case would be the same. However, if you look at the variance, this describes what is the variation band of our signal. And of course, if these are moving, so we are always considering that the molecules are moving around, the intensity that you measure in that pixel will jump more if you have less but brighter molecules. So the fluctuations are larger. So this is described in this plot here. So the variation of your count rate is much higher for brighter molecules than if you have the dimmer molecule. So here we can already make a distinction. So given two series of equal average, the larger is the variance, the less molecules contribute to this average, and the ratio of the square of the average intensity to the variance is proportional to the number of particles. So this is again how we can get the number of particles or the concentration in our region of interest. So how do we calculate these protein aggregations or measure protein aggregations from those images? So of course, in each pixel, we can calculate the average. So sum of intensity values in each pixel divided by the cumulative of all of them. And then we can also get the variance in each pixels. Typically, the units of brightness are, well, they are related to the instrument parameters, which is pixel dwell time, laser power, PMT gain, and all these settings, which you also have to take into account when you're setting up the experiments. And usually the unit, the intensity is counted at as counts per dwell time per molecule. Okay, so our brightness is defined as the average intensity per the average number of molecules. If you have less molecules and the intensity is high, you have high brightness. If you have low average intensity but a lot of molecules, you have a low average brightness. And in the brightness, in terms of the first and second moment that we calculated, is given by the variance divided by the average intensity for each pixel. And we can also rearrange this formula to get the average number of molecules as the average intensity squared divided by the variance. So these are the parameters that we have: variance, average counts, apparent number of molecules n, apparent molecular brightness, capital B, and the number of frames that we analyzed. Okay, so one question could be, how do I set up the instrument? We already said usually the data you get from rigs is pretty good, pretty well suited for number and brightness analysis. But if you're not doing rigs, what should I look out for? Well, again, we get these we get the information about the aggregation state, assuming that our molecules will be able to move while we're doing the measurement, which gives us these intensity fluctuations in each pixel where we can apply our statistics to. So this means we have to have a dwell time that is short compared to the motion of the molecule. So we are able to see the fluctuations. So this is illustrated by the correlation function here. If we are measuring short dwell times, we are able to see the fluctuations. So we have a non-zero correlation amplitude. But if we are too slow, if we average too much, we will not be able to measure these fluctuations. So in this case, the correlation function is 0. So this is something to look out for. General, for the raster scan, we don't have much of a problem because the pixel dwell times are typically always in the microsecond, couple of microseconds, maybe tens of microseconds. So we are fast enough for diffusional motion in cells and tissues. But if you use a camera, you have to be careful to not make your frame exposure time too long. What you can do there is use some kind of stroboscopic illumination. So even you expose longer, you just give a flash of light, very short, maybe 100 microsecond or something. And then, yeah, you will also again be able to see those fluctuations. So this is important if you are trying to do number and brightness on your camera-based microscope, like your TURF instrument or light sheet or something else. Okay, so what is the variance that we see? Well, we have a variance due to the particle number fluctuations. So the particles move, and this makes the signal fluctuate. And this is equal to the brightness of the particle squared times the number of particles that we have in that observed volume. But then we also have a variance due to detector shot noise. I mean, you probably have seen, if you take, even you have a perfectly static sample like a calibration slide or something, you will still see variance in the signal, which is just the shot noise from your detector. And this is, in the case of the number and brightness measurement, this is brightness times number. So the measured variance contains these two terms: the variance due to the particle number fluctuation and the variance due to the counting statistics. So our variance consists of those two terms. And they have different dependence on the molecular brightness. One is dependent to the true brightness squared, and the other is not. This is for the photon counting detector, by the way. So the ideal case for the analog detection, you have to do some more corrections, account for more things. You'll see in a bit. So both parameters depend on the intrinsic brightness and also the number of molecules. So back to our brightness formula, this was defined as the variance divided by the average. Then the variance we just determined is made up of the molecular number fluctuations and the detector fluctuations. We can insert those formulas from the previous slide. And then for the detector, if it's like photon counting, then it's a perfect, it's a Poissonian distribution. And the property of the Poissonian is that the variance is equal to the average. So this term just becomes 1. Here, the epsilon goes out, the number of molecules goes out. So our apparent brightness is the true brightness of our molecule plus 1. This is important. So basically, if you do a measurement of something immobile, like a calibration slide or something, and you do the analysis, you should be getting an apparent brightness of 1. Because, yeah, the true brightness, there are no moving molecules, so this term would be 0, so you should be expecting 1. This is a good way to, if you're doing the measurement, it's a good way to test if your calibration is correct. If you have a measurement of something immobile, you should get 1. Okay, and then you can rearrange the formula to get the true number of molecules n and the true molecular brightness. So that would, for example, correspond to a monomer. And then from that, a dimer should have double the true brightness, a tetramer quadruple the apparent brightness, and so on and so forth. If you calculate it this way, so directly to the true number and true brightness, however, there can be a problem if you go to regions where particles are immobile, because then we don't have particle number fluctuations, so we don't have a true number. You can see that if you're going to a Poisson, this term will become 0, so you will not be able to reliably do this calculation. So we usually use these in our plots, the apparent number and the apparent brightness, so capital N, capital E. Okay, as you've seen earlier, we have this quadratic dependence on the brightness of the variance on the particle brightness. Here is an experimental verification. So this was a 20 nanomolar EGFP solution. And to increase the brightness, you can also just increase the laser power, which in terms tells you to not change the laser power while you're doing the measurement. Yeah, otherwise you're changing the brightness of what you measure. But here we're doing it on purpose just to check. And you see we get this quadratic behavior in the increase of the, in the variance as a function of the intensity that we would expect. Again, this was done using photon counting detectors. Okay, so some more evaluation of what you expect in the measurement and what's the difference between mobile and immobile molecules. As pointed out, the immobile molecules will not give you number fluctuations, and we also get an apparent brightness of 1, independent how bright our sample is. For the mobile, this will be different. So here on the right-hand side, we have our apparent brightness as measured. And then here we use the constant laser power, but increased the concentration for an EGFP solution. These are the red dots that you can see here. And you see that the brightness is larger than 1, which is good. As we further increase the concentration, you see the count rate increases, but the brightness will stay the same. Because, yeah, we did not change the laser power. We only get more signal because we have more molecules, but the brightness of the molecules is the same. And we get this result. We get what we expected. if you push more, you see that at some point the brightness drops off. At this point, you see we're in the region of more than 1 million counts per second. So at some point you will start to saturate your detector, get in the nonlinear range. So you have to make sure to avoid this range. For photomultiplier detectors, the maximum count rate you can usually get is maybe 10 million counts per second, but at that point you're already in the close to saturation, so you should avoid that. Typically stay below maybe 1 million counts per second or something like that. Again, it depends very much on your detector. If you use an avalanche photodiode detector, these have much lower saturation rates, so there you have to be even more careful. Maybe you can only have a couple of 100,000 counts per second. Also in the plot here is shown you a slide where dye is immobilized just in plastic, so the molecules don't move around. So we have this apparent brightness of 1, which we expect for an immobile fraction. And as you can see, in this case, we cannot change the concentration, but we can change the laser power to make it, to have more intensity, more counts. And again, you can see it does not change until you hit this kind of saturation here. Okay, so how about something a more real sample, or more complicated sample, I should say. The other one is still real. So here's a measurement of fluorescent beads immobilized on a glass surface, but there's also added 100 nanomolar fluorescein dye solution on top. So this is the intensity that image that you get. Here you see these beads, immobile aggregates, and also some intensity from the fluorescein, but this is much lower, much less intense than the beads. If you calculate the brightness map, you see that it's kind of inverse, because in the calculation, the immobile fraction has a brightness of 1. So these are these bluish color codes here where the beads are. And then from the molecules that can move, you have the additional component from the fluctuations from the movement. So we have this higher apparent brightness here. Here we have it quantified. So this is our plot of the apparent brightness as a function of the intensity for every pixel. So this is a 2D histogram containing all the pixels of this image here. So if we want to select the fluorescein, we select a pixel that have brightnesses larger than 1, because this is the addition from the number fluctuations. And if we do so, you see we highlight this portion of the image where we have the fluorescein. If we select all the pixels that have a brightness value of around 1, you see you highlight the immobile fraction, which in our case are the beads. Here is a series of different aggregates from monomer to octamer. Again, if it would be immobile, you would be expecting 1. Then the monomer has apparent brightness a little bit more than 1. And then the dimer has double of that. The tetramer 4 times of the monomer, octamer 8 times of the monomer, as you would expect. Okay, here's some more control measurements. This is again EGFP, monomeric EGFP in solution. Here is our 2D histogram. Again, brightness versus intensity. And again, you can see that the center of mass of this cloud containing all of our image pixels is a bit above 1, with the difference between 1 and this average being the brightness of our green fluorescent protein monomer. Of course, we have noise, and so it's a little bit of a cloud, and yeah, there's some uncertainty in the measurement. But on average, you can very accurately get the brightness of the EGFP monomer. Okay, so what happens if we plot the brightness versus laser intensity? Of course, if you increase the laser intensity, you will get more fluorescent photons per molecule as expected. So again, if you do the measurement, put on your cells and find a good laser setting where you can get a decent signal from your cells, but then keep it at that. Because if you change it, your data will not be comparable. The same is true for the gain of your photomultiplier. If you choose that, you will get different values for different measurements and you can no longer compare them. So you have to be sure to keep all those parameters consistent during the measurement. Here, just as a control, what happens if we increase the concentration of the EGFP solution? As expected, the observed number with that we get with the number and brightness method also goes up. And as we increase the concentration, the brightness stays constant, again, as expected. If we just increase the number of molecules, we don't increase the brightness that we measure. Okay, this all works very nicely for true photon counting systems, but what about systems that are working in the analog mode? So we have to have additional considerations with analog detection systems. So one is that we record the digital levels instead of photon counts. So if one photon arrives at the detector, this can give us whatever 10 digital levels or something, or maybe 20 digital levels. So there's some conversion factor there that we have to determine. In addition to that, there's often an offset. So even if you have no photons coming on your detector, you still are getting some digital values. And then in addition, we have noise because of the amplifier of the detector, especially at low currents. Okay, so we have these three parameters that we have to determine: the offset, the analog offset, and this S factor, which converts digital levels to photon counts. And then we have to consider also the variance or the noise of our analog detector. Yeah, and then once we do all these, we measure all these parameters, we can enter them in the software and correct all of these to get an accurate result. So here, just to show you what does a typical analog detector response look like. So this is a histogram of the digital levels that we find in all the pixels. And you see we have, first of all, we have this offset here. So it doesn't start at 0. So this red dotted line corresponds to the detector offset. Then we have this peak here, this kind of Gaussian distribution, which is our amplifier noise. And then we have this exponential decay here. The y-axis is a logarithmic scale, so this is why it's a line here. Corresponds to an exponential. And depending on the slope will describe this conversion factor from photon counts to digital levels. So you can measure basically the dark from your detector and then go in this calibration page where you can determine all these three parameters that you can then use in your number and brightness analysis. You'll see that on Friday. Here, just to show you in comparison, this is a photon counting detector. And here you can see that the occurrence of pixels that we get with 0 counts, if we have 0 intensity, so no sample on, no laser on, almost all of them are 0. We have very few. Again, this is a logarithmic scale, so in this case we have 1 million that are 0, and then only maybe 150 that are 1, and then just a couple of pixels higher than that. So this is, if you have a photon counting, a true photon counting system, this is very nice because you don't have to deal with these additional parameters to correct for your detector and electronics. Okay, here is an example of a 20 nanomolar EGFP in solution. Again, we measure the apparent brightness while increasing the laser power, so perfectly linear behavior if you don't saturate the detector. And the same is true for the number of molecules. As we increase the laser power, it stays the same because the measured intensity only increases. Sorry, the number of molecules does not increase. We only increase the signal because we increase the laser power. So if you do all these corrections, the recovery of relative values is very good in the analog system. For absolute values, you have to be a little bit more careful. So maybe do a calibration measurement where you know the concentration of your solution to get an idea. But anyway, often in cells, you're interested more in relative changes. So did it double, did it triple, did it quadruple? And for this, it typically works very well. So here's an example of a cell. And in this case, it's just expressing the green fluorescent protein, which is not expected to form aggregates. But you can see we have if you just look at the intensity image, we have a gradient of the average intensity, because in this case, we just happen to have more molecules here towards the inside of the cell than on the periphery. So if we plot the map of the number of molecules, we can see this gradient in the concentration. If we map the molecular brightness of the molecules, you see it's uniform. It's the same everywhere because this EGFP did not aggregate in the cell. It's uniform throughout the cell. Okay, so what can you study with this method? Again, we are looking at the focal adhesions, the Paxillin molecules, and in specifically during migration where these adhesions assemble and disassemble. So the hypothesis is that adhesions mature by increased associations, which leads to these aggregation of molecules and protein density around the adhesions that form. So how can we quantify this? Well, we can use the number and brightness method. Yeah, again, if we have many dim molecules, we should get less strong fluctuations than we have fewer bright aggregates, provided those aggregates still move. So here is an image of a cell expressing EGFP Paxillin. You can clearly see those focal adhesions where you have very high intensities. So this is the average intensity map on the left. And then we have the brightness map on the right. And you see we have kind of lower brightness values, so indicating monomers here in the cytoplasm. And then we have some high brightness kind of near the adhesions. Okay, let's go to this data in a little bit more detail. So here we have our NMB plot again. So apparent brightness on the y-axis, intensity on the x-axis. Here with this this kind of pink rectangle, we select a monomer. So this is the brightness slightly above 1 that corresponds to the EGFP monomer for this measurement. And if we select those pixels and highlight them in the image, you see all those pixels come from the cytoplasm area. Now here in the bottom row, instead of the monomers, we only select pixels that have a very high variance, so very high brightness values. And if we map those back to the image, you see that regions around the adhesions get highlighted. But interestingly, it's not necessarily on top of the adhesions. It's often more around the adhesions. And the reason is, once the molecules get incorporated in those focal adhesions, then those molecules cannot move anymore, which means they become invisible to the number and brightness method. They just become this apparent brightness of 1 as an immobile fraction. But these larger clusters, while they integrate, they still move. And this is why we find these high variances near or around these adhesions, and only as they form or disassemble. So for example, when the cell moves. Yeah. You can also look at the histogram where you have these different populations of the brightness. Here, fitted with the green curve is the monomer, and then we have the higher brightness ones, which corresponds to these larger aggregates. And we can estimate the average cluster size from this. And for this measurement, it turns out to be about 8 Paxillin molecules that are in these clusters that then assemble into this bigger adhesion complex. You can also do time sequence, which is very interesting. So, for example, here we recorded 500 frames and split them up into smaller chunks of 100 frames that we analyzed individually. And then, for example, in the first 100 frames, you can see we have these high brightness values, so lots of these complexes that have formed, and then with ongoing time, these larger aggregates start to disappear. Or they were integrated now into the focal adhesion. You can also see that as a function of time, the average count rate was going down in this case. Then it's constant here while the formation has completed, and here it was going up again as the cell was migrating. Okay, so to summarize, the number and brightness analysis can distinguish between the number of molecules and the molecular brightness or the aggregation state in the same pixel. So here we really get a pixel resolution map. The acquisition of number and brightness data or data for NMB analysis can be done on laser scanning microscopes. You can use the same data you acquired for RIGs, but you can also use camera data with the caveat that I mentioned earlier, which was that you have to make sure your exposures are very short, or you just flash the light to make it very short. Immobile fractions can be separated out because they have the apparent brightness value equal to 1. And I've shown you that the number of brightness analysis of Paxillin was showing that we have this clustering of molecules before they form these larger adhesion complexes. One thing I wanted to point out in the RIGs experiment, we were selecting a smaller pixel size to be able to fit the correlation function. In the number and brightness, we are not doing a fit. We're analyzing each pixel individually, just doing statistics. So you don't have this constraint for the pixel size. So if you're doing a measurement where you don't want to do RIGs, you're not interested in the dynamics, you're only interested in the number and brightness, you can select if you want, you can select a larger pixel size to maybe be able to image the whole cell and not just a small region of interest. But then you cannot analyze this data with RIGs. Of course, I wouldn't make the pixel size too large, because then, of course, you lose spatial resolution and lose detail of your measurement. So often for measurements, if you have a pixel size that's kind of around the size of the point split function, yeah, if you want more detail, you can zoom in a little bit more. But again, for the RIGS experiment, for the number and brightness experiment, you're flexible in the pixel size. But then again, remember, for the number and brightness experiment, you're not flexible in terms of setting of your laser power, PMT gain, and other things that may change the signal on your detector. You sort it out in the beginning of the experiment, and then you stick with your settings throughout the entire experiment and do your calibration based on that. Okay, again, with